is working?
It should be working.
No.
No?
No.
Maybe you can use mine. Can you make a try again?
What?
Can you make a test? Say something?
Yes. Hello. Hello.
Hallo? Anscheinend funktioniert das Mikrofon nicht.
Hello.
Das Mundmikrofon.
No.
How do you maximize
I may
that?
try with it. Oh,
For
now
now.
it's working? Hello. Hello. Sorry, I don't speak German.
Okay. Can you maybe speak in English so that the speaker can understand you?
Hello.
That's the right slideshow, no?
Yeah, exactly. Yes.
Just because when you talk with your colleague, I don't open the mic. Then you start.
Okay.
So, it is working now.
Yeah, try.
Okay. So, first of all, after Professor's clip presentation, I changed my title a little bit. So, one day we got hopefully wholesale models. But, well, we believe that roughly all the necessary information for a cell to grow and reproduce itself is based in its genome, one-dimensional information. From the sequence of nucleotides, we can extract a second dimension of information like genes and promoters, and also another sequence of interest. And we know that cells use this information to produce RNAs, proteins. There are molecules that interact within themselves through thousands of physical and biochemical reactions. But also, they interact with other smaller molecules like metabolites. So if we could put together all these interactions among molecules we know inside, that are inside a cell together, what we have would be a map where all the cellular processes are naturally represented by their underlying biochemical reactions and also naturally integrated because of the interactions among these molecules. So, as an example, we did that for an organism called Mycoplasma genitalium, which we have a database with enough information to do that. And in this model, what you have is a network with nearly 2,500 molecules and 4,000 reactions, where we could represent all the known process about the cell, which encompasses several kinds of molecules. So the question now is, how can we extract dynamic information or behavioral information about this organism from this map? So I'll briefly introduce our approach that we are working on. And basically, we can write down these networks into a mathematical model by writing it down as a stoichiometric matrix. We can also set to the system some constraints like mass balancing constraints. And then we can say that it behaves like a mechanistic model where concentrations evolves in time through heights that are dependent on the previously concentrations. To simulate that, we proposed an algorithm called constraint-based simulation algorithm that basically aims at do scalable simulations while keeping account for discrete concentration, number of molecules, the stochasticity of the system, and still being computationally efficient. Well, we know that estimating rates for systems that are really big is way difficult and requires a lot of parameters, kinetic parameters. And we are trying to do is to use diffusion-based models in order to reduce the number of kinetic parameters for the system, and also making the system responsive to global physical parameters, such as temperature and medium viscosity. So the main goals of our project is to have simulation methods that are easy to model, that have an homogeneous representation of cellular processes, so that we can use this information to make scalable simulations. Well, if you're more interested in this work, we can discuss after in the poster section, because I just have 5 minutes. And I would like to thank you. Thank first ISBC for helping me in many ways to be here today, and also funding from CAPS. And thank you for the audience.
So thank you very much. Do we have questions? Okay, so I was wondering how your model depends. Your model depends on knowing the regulatory network of a particular organism. So I wonder if you want to use this approach in a different organism, how dependent is you? You need to have a new regulatory network for the new organism, or can you somehow infer from one about regulatory interactions?
Okay. Well, in these models, what we want is to represent cellular processes based on their underlying biochemical reactions. So regulation processes are within these reactions. Like we have regulation through protein binding and other signaling molecules. So what we want is not have separated models for separated processes, but model in a way that they are already integrated before simulation.
Okay. Any other question? Okay. Sorry, one question there.
Yeah, thank you. Just a curiosity. So as far as I understand, then you're solving partial differential equations for the diffusion model, right?
No.
You're not?
No.
Okay.
Actually, we're using this constraint-based approach where we can discretize the space and then calculate fluxes between the discrete spaces.
Okay. What is your poster number, please?
7.
So, thank you.
Okay, so let's thank the speaker again.
Thank you.
And our next speaker is Christoph Leberecht, which comes from Hochschule Mittweida, Germany. And